Good morning, students. Welcome to my channel. My name is Dagu Shanibulu. Now, today's recap is on STH 313 microbial immunochemistry for students of polytechnics in Nigeria. And for my students who are coming here, we had discussed most of all these in class. I just want to go over it a bit and just discuss just the peripheral as you plan and going into the test. Now, when we resume, we explain that the course is all about helping you understand the chemical aspect of microbial immunity. And first of all, with this, when we understand, we first of all discuss the structural component of a microorganism. Prior to this, we discuss organisms that are regarded as microorganisms. We mentioned some of the kingdoms that constitute microorganisms. We talk about the monoid kingdom that is part of it. We also mentioned the protozoa that are also part of this, and then some fungi also fall under the microorganism category. But we discussed the structural component of protozoas. We say that the architectural component is made up of three structures. We have the appendages, we have the envelopes, and then we have the cytoplasmic region. The appendages we say include flagellas. They are also the fimbriae and the pili. The envelopes include the capsules, the cell membrane, and the cell walls. Then the cytoplasmic region, we talk about the various components that are found there. We say in the bacteria, inside the cytoplasm, they have a nucleoid, and you also have some, beside the cytosol, they also have some inclusions that serve as storage structures for various food substances and also ways of disposing some of the substances that they have in them. And then after that, we discuss the various functions of this as it relates to microorganisms, and then we also discuss the microbial growth. We say that the microorganism, the life cycle of a microorganism is represented on a graph known as the microbial growth curve. Say that the life of a microorganism starts, the very first stage is called the lag phase, at the stage where they are trying to get used to the environment that they are colonizing. Then we have the log phase, where there is a rapid growth. It is at this stage that research are carried out. It's at this stage that the generational time of microorganisms are known. Then we have the stationary phase, where the rate of the death rate and the growth rate of microorganisms is not well defined. As you know, there is no growth. More are dying, so there is a balance. And then there is a death phase, the death stage, where there's a decline in the amount of microorganisms that are found in a particular culture. And we say that the rate at which microorganisms grow, when we talk of growth in microorganisms, we are not talking about growth in size and in shape and in height. We are talking about the multiplication in number of cells. And we say that the time that it takes for microorganisms to the population of microorganisms to double is known as the generational time. And generational time is given as G is equals to T all over N. T is the time taken for the doubling, while N is the number of generation. And that was that. And we calculated G. We say that we calculated it to be given as G is equals to T all over N. And then we discovered that N, this is given as T all over, and N itself is given as log B. B is equals to so we say the life cycle of a bacteria is described on a graph known as the bacterial growth curve. The bacterial growth curve looks something like this. We have here is called the lag phase, here is called the log phase, and here is the stationary phase, and here is the death phase or the decline stage, the decline phase. Now at the log phase, the bacteria are trying to get used to the environment. They are trying to grow. They are trying to get accustomed to what is happening. They call the medium that they are growing in. While at the log phase, they have gotten used. Now there is rapid growth. Then at the stationary phase, the reproduction of bacterial cells is offset by their deaths, and the population reaches a plateau. Now it is at this stage that secondary metabolites are developed by bacteria. It's at this stage that they develop antibacterials, they develop spores. Spore formation is formed at this stage. Now after that, we discussed the growth requirements for microorganisms. We say that some of the requirements that microorganisms need, they are physical, some of them they are chemical in nature. So we mentioned some of the requirements that microorganisms need. We say that after understanding the growth curve of microorganisms, we also discussed the microbial measurement. How do we measure the growth of microorganisms? We talk about some of the steps that are involved. We talk about the plate count method. We also determine the most probable count number. We also did a direct microscopic count. We also discussed the turbidity method of calculating bacterial growth. We also discussed the dry weight of using the dry weight of a culture to know the amount of microorganisms that are there. And then we discussed the microbial cultivation. How do we grow microbes in the lab? Say that the process of growing microorganisms in the lab is known as culture. And the microbial culture can be either a pure culture or a mixed culture. Now, the medium on which microorganisms are cultured is known as a medium. Now we have different types of media. We have the general microbial media. We also the special microbial media. We also have the differential media, and we're able to explain each and then we talk about method of isolation of microorganisms. We discuss the method of isolation, including the strict plate method. We also discuss the pour plate method of microbial isolation. Then we discuss the growth requirement for microorganisms, where we say that some of the requirements are physical and some are chemical. The chemical method include all the minerals and all the elements that microbes need to thrive. We say that some microorganisms are called aerobic because they require oxygen for their survival. Some are called anaerobic. And after that, we see that some can thrive both in aerobic and anaerobic conditions. So we call those ones facultative and microorganisms. Then there are those that require just little amount of air. Those ones are called microaerophilic and microorganisms. And then the physical requirements say that there are some that thrive in under extreme conditions, like a very extreme cold. Those ones are called the psychrophilic and microorganisms, or psychrophile. Then we have those that thrive in a temperature from 0 to 20. Those are called the mesophilic microorganisms. And then there are those that prefer a temperature from 20 to 40. Those ones that affect us, we call those ones the thermophilic organisms. I will go over that again. We say that there are other organisms that are called sacrophiles. They prefer a temperature of 0 to 20 degrees Celsius to survive. Then there are those that we call mesophile, those that can survive on a temperature that's from 20 to 40 degrees Celsius. And then there are those organisms that we call thermophiles, those that can survive in a temperature from 40 and above. Other physical factors that bacteria require for their survival include the medium. Either the medium is acidic or basic, or it is a neutral. So those that thrive under a neutral pH, a pH of 7, we call them neutrophiles. And then there are those that would prefer acidic medium, Those that prefer acidic medium, those ones we call them acidophilic microorganisms or acidophiles. And then there are those that will thrive in basic conditions. Then some microorganisms thrive better when the pressure is ideal, not just. Then we also did calculation on how to determine the generational time of microorganisms, and we did that. The formula that was written on the, like we said earlier, generational time is given as T all over N, where T is the time that is taken for the generation, the doubling of the microorganism, and N is the number of generation. And we, based on them, we utilize the formula B is equals to I times 2 raised to the power of N, where B is the final population of microorganisms, while I is the initial population of the microorganism at the beginning, times 2 raised to the power N, where N is the generation, the number of generations. And from there we solve, and we discover that N is equals to 3.3, the logarithm of the final population of the microorganism all over the initial population. This gives us the general formula of G is equals to T divided by 3.3 log of B all over I. That is the general formula for the calculation of generational time of microorganisms. After that, we discuss classification of microorganisms based on their nutritional requirement. Now, there are 3 basic classifications of microorganisms based on their nutritional requirement. The nutritional requirement could either be their source of energy, could be their source of electrons, and could be their source of carbons. So for source of energy, we have 2 basic types of microorganisms. We have the phototrophs and the chemotrophs. The phototrophs utilizes the sunlight for their energy, while the chemotrophs generates their energy from chemical reactions like the breakdown of food substances or organic matters to get their energy. Then, based on the source of electrons, we also have 2 groups of microorganisms. We have the lithotrophs and the organotrophs. The lithotrophs are organic or bacteria or microorganisms that reduce inorganic compounds and generate electrons from there. While organotrophs, they use organic substances as electron donor. Organisms that utilize this includes the E. coli. Then, based on their source of carbon, we have 4 different types of microorganisms. We have the chemoautotrophs, we have the chemoheterotrophs, we have the photoautotrophs, and we have the photoheterotrophs. Then we talk about their sources of nutrients. Chemical sources are carbon. The chemicals that microorganisms need include carbon, nitrogen, sulfur, phosphorus, oxygen, trace elements, and organic growth factors. And where do they get? You know that carbon is the structural backbone of all living organisms. So chemoautotrophs, just like we have talked about up there, they get most of their carbon from organic sources such as protein, lipid, and carbohydrate. Fermentative microbes like yeast convert glucose, which is a monosaccharide, to alcohol. At the same time, they release energy that is used to power their metabolic activities. In lactobacillus and lactococcus, glucose is converted to lactic acid and energy is released in the process. The amount of energy generated by these organisms is low compared to aerobic microorganisms. And maltose, sucrose, and lactose are made up of 2 glucose units. That is, maltose is made up of 2 glucose units, and sucrose is made up of 1 glucose unit and a fructose, while lactose is made up of 1 glucose unit and galactose. So microorganisms break down these various sugar and disaccharides when they break them down, they release glucose that is used to generate energy, and the process that they do this is known as saccharification. The glucose component is used to power up ATP and as source of carbon. For polysaccharides, and microorganisms utilize a group of enzymes called cazymes, which are known as carbohydrate active enzymes. Nitrogen source are utilized for biosynthesis of nucleic acid, and nitrogen are taken up as nitrates or from reduced organic nitrogen such as ammonia salt. Organic sources of nitrogen include amino acid. Organic nitrogen sources support rapid growth and high cell yield of bacteria than inorganic sources. An increase in nitrogen sources has a bad effect on gram-negative bacteria. You know that gram-negative bacteria utilizes phosphorus to form the phospholipid that surrounds them. So if there is high nitrogen, phosphorus will not be available. Another way nitrogen can be sourced by microorganisms is by nitrogen fixation. Although not all microorganisms carry out fixation, the organisms that carry out fixation include rhizobium, that is found in the root nodules of leguminous plants. Then phosphorus is utilized by microorganisms in the formation of an ATP, and some of them utilize it for the formation of their phospholipids, like gram-negative bacteria. Then sulfur is used for the synthesis of sulfur-containing amino acids like cysteine, thymine, and biotin. Sources include sulfate ions and hydrogen sulfide and sulfur-containing amino acid. Now, that is how far we have come for this class, and there are other things that were not mentioned, but for the sake of this test, we have done the recap. And for the test that is coming up, the link is below. Click on the link, and then the page for the test will be open for you to participate in it. Now, have a nice day and good luck in your test. Bye.